out some claim of our independent panel of experts.
Please take your seats in the back. We're about to get started. You'll have to model it for me. Okay, welcome back, everybody. We are kicking off our afternoon session with a couple of conversations around the theme of health and life sciences. Just a reminder for those who may be tuning in online, this is State of Innovation, Big Ideas for the Future. Day one project, we are a project of the Federation of American Scientists. Excited to be launching today with a series of catalytic conversations and kicking off a year-long agenda to identify 100 inspiring science and tech policy ideas. Our next conversation is titled Frontiers of Bioinnovation. And we have Mahlet Mesfin, visiting scholar at the Penn Biden Center for Diplomacy and Global Engagement, leading the conversation with Eric Lander, who is the president and founding director of the Broad Institute of MIT and Harvard. Please join me in welcoming the two of them to the stage.
For the organizers for pulling together this conversation. I'm really honored to be here with Dr. Lander to talk with you guys about an issue as a bioengineer that's near and dear to my heart. We heard earlier today from Dr. Holdren about how this entire project sort of encapsulates the interdisciplinary nature of these issues and the intersectoral sort of approach that's needed and the interconnectedness between the national security and the economic and the environmental issues. And I think bio as a topic, and innovations in bio really encapsulates sort of all of why those things are important. And just talking about the bipartisan nature of this issue, there was recently, just 2 weeks ago, a bill that was introduced on industries of the future, and you talked about AI and wireless, quantum, and SynBio and biotech were a part of this component. So this is an issue that's important in the policy space. It's bipartisan, and it's a dynamic space, and we're really excited to have sort of Eric Lander give us some of his thoughts, not just on the technology, but also as someone who's been a part of and adjacent to so many of the policy and the advisory boards and processes around this issue over the last several years. So thought we'd think a little bit about the past and the present and the future and interweave all of that together. So, and looking back, if you look over the past decade and thinking about the life sciences and biotechnology space, what are some of the surprises in terms of like where we're here now, where you thought we would be, where we're not, where you thought we might be there, and can you think about the government's role in either being there or not being there?
Yeah. So to put that in the right context, I might go back a little further because I think that really deepens what the surprises have been. So my own scientific career in the life sciences is pretty much coterminous with the start of this Human Genome Project. And I think that was a really interesting shift. The idea that biology was like finite, bounded, about information. We didn't really understand that in the late 1980s when the Human Genome Project was starting. David Baltimore, one of the great scientific leaders of his generation, building the Whitehead Institute, made no provision for a computer in the Whitehead Institute. And then at the last minute, they put a computer room in the building to have the computer. And that sort of marked the time at which that was all kind of shifting. That's when data referred to stuff you wrote in pen in your notebook. And when the probability that when you were writing a paper, it cited anything other than something generated in your own lab was a rare event. I think over the course of that decade and going into the early 2000s, we began to think about biology very differently. There were only 3 billion bases in the human genome. We could know them all. There are only 20,000 genes. We could know them all. Now, of course, students ask me in my lab, how did you ever do anything before you knew that? How did you get anything done? Because they take it for granted. And some of them imagine, well, it must have been done in the 1950s or the 1800s, because they can't imagine doing biology without that. But what's the real surprise for me is how much we underestimated how much legs this would have. Because in the early 2000s, we were done with the Human Genome Project, and then people said, okay, fine, we can go back to business as usual. But no, very quickly, we had one human. We needed to know all the variation in the human population. And we had to understand how it correlated with disease. And today, millions and millions of people have been analyzed at the level of genetic polymorphism, allowing the mapping of hundreds of thousands of regions underlying lots of diseases. But it wasn't very long ago, like 2007, when the number of discoveries of that sort you could count on the fingers of maybe fingers and toes, possibly, but you wouldn't have to go any further than that. And then it just like skyrocketed. But then again and again, cells, the cells of our body. It's only in the last 5-6 years we've come to understand that we have lots more cell types than we ever imagined. But now there are methods on a single cell basis to read out the genetic programs of all these cells. Those methods got developed, and within 3 or 4 years, there was an international human cell atlas involving 66 countries trying to generate all the data on all the cells from lots of different kinds of people. And then it goes on and on, that the idea that we can build systematic knowledge as a public good, there's already young people who are thinking crazy thoughts like, okay, it's good there's a finite number of genes and a finite number of cells. Are there a finite number of programs that the human genome knows how to run? Could we define all those programs? And they're thinking about methods to discover like the whole catalog of cellular programs. And then that's because, of course, they're bringing in machine learning. They're all becoming fluent in machine learning because they know that they're going to do that as an inference problem from massive observations of data. Then we want to turn our healthcare system into a learning system. We're going to have to make observations. So we can go into some of those in more detail, but the key thing to say is there turned out to be so much more power in thinking this way, and these turned out to be inherently about public goods. And I think the US government gets huge, huge credit over the course of multiple administrations to sustain the rigorous thinking about where can we build such public goods and how do we do it efficiently. Remember, it's really important to not do a whole lot of things. So not every idea like this is a good idea, but there's been a succession of really good ideas. And the government's been there to say, yeah, we should figure out how to back it. Now, we're not unique. The UK's done a good job. Occasionally, the UK's done better than we have. Occasionally, we do better than them. But I'd say those two governments have played an incredibly important role. And when people ask, what do we need government for? What do we need public action for?
I was going to ask that. What do you think the government did well then to be able to?
Oh, I mean, this could never have happened without some organizing function of all of it, because all of these projects were greater than any individual lab. They were greater than a company because they weren't a business plan. They were public goods. They were the information that could drive 10,000 businesses. They were the information that drive 10,000 labs. But in the classical economic definition, no private actor could truly pay to do this because no private actor could appropriate all the fruits of it. This was something where the government said, we're doing a human genome project, for example, just to pick on that one, and we're not worrying about how we're going to, what the business plan is to make money. The business plan is, it's going to teach us how disease works, and that's going to drive tremendous progress in making therapies and other things. And there were, at the time, 1 or 2 private efforts to try to do that, and they didn't work because a private effort wasn't matched to building a public good in this way. I mean, they got stuff done, but it didn't work as a business. I think that foresight to understand what are those things that we can only do by working together has been an incredible success in many, not quite all areas. We've screwed up a couple of them we could get to, but in many areas, I think the US gets tremendous credit, and it has gone across administrations and parties. The Human Genome Project was started, for the first couple of years under George Bush, who announced it in the State of the Union. Through not ideal staff work, it was announced as the Human Genome Project. But in any case, it improved past there, and President Clinton, and then all the way, it's just been going on, and under the Obama administration, and under the current administration, we're seeing investment in these sort of things. It's great.
So you touched a little bit about sort of this difference between the public good, sort of within the government, and sort of maybe more commercial interests in industry, and sort of there's all of these parts of the ecosystem that really need to work together. And so you've again been a part of so many advisory boards and conversations about bridging those gaps between the communities. Do you have any lessons learned that you?
Well, it's really important, as I said, to understand what the government can do. It's also important to understand what industry can do. Because in the biosciences, there are a whole lot of things that are private goods that government really isn't ideally suited to do. You want to make a drug, you really have to invest hundreds of millions of dollars. Nobody's going to do that unless, at the end of the day, they actually have some protection, some limited monopoly to be able to realize that value. So I think we want to think very carefully, what things should not be patented? Information should be put out there. What things must be patented? Because no one will ever invest in it unless they can get protection. So understand, I sometimes just refer to this amazing thing as the miracle machine. The government, academia, and industry. And we have this incredible flywheel in this country. And we understand what those pieces do, and each piece does a part the other doesn't. So in making therapeutics, there are roles that the government or somebody like the government should be doing right now, which are to create general methods. I see therapeutics changing in interesting ways. I think 30, 40 years from now, the students looking back will say, oh, how quaint that you put little molecules in a bottle and spent your time figuring out how to adapt, put a methyl group here to make it bind a little better, and every individual drug was its own special little experience. I think we're going to think about programmable therapeutics. We're going to think about how do we deliver a drug to this cell type, but not other cell types, which is going to open up what they call a therapeutic window. It might be a drug that has a good effect in this cell type and a bad effect in several other cell types, but we're not going to deliver it to those cell types. So suddenly we get to be able to be much safer and more efficacious. But it means we need like a post office that delivers things to designated cell types. That kind of thinking is a public good thinking. The use of it is going to be in 1,000 private projects. And industry and academia need to think together, because the government alone trying to do that without the experience from industry won't work. But industry trying to do it, what will happen is they're going to focus on how do I deliver to this one cell type to make a drug that I can make a return on today, and not reap the benefit of building the whole thing. Same thing for programming. We now have CRISPR, which can direct proteins to particular genes. Well, with CRISPR, you can begin to write programs. Turn on gene A and B, if not C. And you might imagine eventually kind of the C++ code that kids write today will be matched by a D++ code written in DNA, where you can write programs to do things and be able to predictably say, I will be able to turn down this pathway in this way and turn up this thing, and it's not going to be so infinitely unpredictable. It's very hard. People still have a hard time imagining, but if there are a finite number of cellular programs, for each disease, we're going to know the cellular programs that are disrupted. We're going to know the genes that are turned on and off. We're going to know, to quote the geeky word, the transcription factors that turn on those genes. We're going to actually be able, at some point in this century, to be able to write code to do those things. But building a world like that is going to involve public investment and private investment. And look, what excites me about being involved in public policy here is that I see all sorts of things that I know wouldn't get done without good public policy.
So you touched a little bit on this in general, but when you think about sort of biotechnology as an enabling technology with multiple different applications, and I think you think about AI and machine learning in a similar framework of how you think about it as a technology area. What do you see about the merge in those two areas? And maybe it's related to what you were just saying about sort of understanding the different cells and sort of going through the data and being able to process it in different ways. But what do you think the opportunities are?
It's a deep question you're asking, an important one. Again, it just shows how naive we are. When we were beginning this journey, we thought, well, as biologists, we'd collect all this information. We'd need some computer science to help us out. now it's becoming the case that the fundamental discoveries are increasingly being made by the people who think in terms of machine learning and computer science, because in the old days, you'd write a paper about this cell type and this gene turning something on and manipulating it. Now what you might do is you might get the whole gene expression readout of 500 cell types under a zillion different conditions in healthy and non-healthy people. And the thing becomes, infer what's going on. Reverse engineer the code, for example. Well, the hard work is actually no longer generate the data. The hard work is infer from the data what's going on there. So we need a whole generation of people coming along who are just utterly bilingual between machine learning and modern biology, because the discoveries are going to be there. There were previous things you discover by purifying something in a test tube. We're now going to purify these programs in a computer, and we're going to need people like it. So, but what's amazing is the incredible scientific community of the U.S., both in industry and in academia, seems to provide. At the institute I run, 5 years ago, we didn't have that many people who were fluent in machine learning. Well, today there's this models, inference, and algorithms meeting that goes on once a week, and 100 people show up from all over Boston to talk about how it all happened. It just stuff nucleates when there's enough concentration in the solution. It nucleates and it precipitates, and it's just amazing to watch these communities grow, because young people, which is of course what we mostly have to continue to back in science, they are incredible. They have an incredible nose for the future.
So, I mean, you were talking about such fundamental shifts in the way that people do science and think about science. And it's not just the U.S. that's sort of thinking about these things, and it's not just the U.S. that has the capabilities. And so, can you think about different approaches of working with other countries and in these areas that are sort of driving innovation forward in biotechnology that works sort of for our interests and also public good?
Yeah, no, very much. It's happily the case that a lot of these public goods, we're not disadvantaged if other countries have them too. I appreciate there are areas where it gets complicated and one wants to beat somebody else or withhold something, and there are good reasons why one might do that. This is an area where, on the whole, most countries have an interest in the fact that all their populations get sick. In the United States, Republicans and Democrats both get sick. So these tend to be issues that unite us. And so, Human Genome Project, 6 countries were involved. This Cell Atlas, 65 countries are involved. And you learn a lot from it. I think I have found that every one of these projects has had international participation, and they've been much better because of them. We just launched a new one on common diseases, and having representation, for example, from South America and from Africa has truly broadened the conversation in important ways. So I think because there's relatively little downside to other people also succeeding, they've been really fertile grounds for international collaboration. Even taking things like in China today, the coronavirus that's in Wuhan.
I was going to raise that.
Well, for example, what are the issues here? I mean, the issues are we're still very poorly fixed for rapidly developing therapeutics. We need to be able to do diagnostics like this, therapeutics like this. Right now, developing therapeutics is a 7 or 8 year journey. But if we're going to deal either with naturally emerging pandemics or, God forbid, biothreats, we need to be able to have things in which we can respond very quickly. And happily, even there, biosecurity and public health are well aligned because roughly we need the same set of tools to support both missions. So, antibiotic resistance, this is another area where we're not well fixed. I don't know that we've done as well as we could, but we need to really focus on antibiotic resistance because it keeps getting worse right now. And yes, public good, there isn't as much private incentive to build an amazing antibiotic that you take for 2 weeks because you can't charge enough for it. we do need, and John and I both worked on this issue when we were doing PCAST. We really do need to think about how to create the right incentives to unleash creativity in industry and in academia to do this. So there are hard problems around these sort of things that have to get solved.
I think we only have 1 minute left, and when you think about the Human Genome Project, which you talked about, there's sort of a time before and after it.
Yep.
When we could sequence and when we can't. CRISPR, maybe lesser extent, but you have a before
Also pretty amazing.
and an after, right?
Yep.
What's the next before and after sort of in the life science space?
Oh my God, if I knew.
Sorry.
Right.
We didn't get a chance to maybe talk about this before.
Yeah. No. It's a great question. It's really tough. These things come out of left field. Take CRISPR. CRISPR was not. It was a 20-year process involving scientists in countries all over the world. There's a lot of deserved attention to the couple of people, including one person at my institution, some other people at Berkeley and in Europe. But there were 20 years of work from people all over that took a shaggy dog story from a Spanish scientist studying some salt-tolerant bacteria, eventually to this transformative technology. And what I love about science is it's this incredible ensemble act where you don't know how the play is going to end. And at every stage, until pretty much the last act of a 10-act play, you would have said, well, this is a curiosity. And then suddenly it bursts on the stage. So it's really hard for me to predict the next one, although I bet it's out in the literature right now, and I'm just not seeing what its incredible impact will be.
I love that analogy about the story, and you don't know the ending, but you have to be a part of the whole process all along. So
Yeah, but unfortunately we do know the ending here because this red light is on.
yes. So thank you so
This
much.
is
for your
wonderful. Well, thank you.